Okay, let me repeat. On the exposure of the allergen, the pre-sensitized mast cells, the receptors, they cross-link with each other, and then they release the biological active substances such as histamine, proteases, neutrophil chemotactic factor, and eosinophil chemotactic factor. Let me talk about histamine. It is a very strong vasodilator. It increases the permeability of vessels by shrinking the endothelium, strongly spasmogenic, stimulates glandular secretions. Proteases, they further leads to tissue damages and more inflammatory mediators are produced. Also, it converts C3 or C4, C5, sorry. These are the cells of the complement system to the active form C3A and C5A. Sorry, the mast cells have receptors for these, whereas these two chemotactic factors, they locally amplify the mast cell, thus increasing the more inflammatory reactions. Another situation may also occur when the pre-sensitized mast cells are again exposed to the allergen. The nucleus is activated with the help of a signal sent by the receptors, and then the mRNA which is present in the nucleus, they release small peptides which are soluble, known as cytokines. Examples of these cytokines are number 1, TNF or the tumor necrotizing factor, interleukins 1, 3, 4, 5 and 6. Here interleukin 3 and 5, they are very important as they are very strong chemotactic agents which for eosinophils, which causes inflammatory reaction. GMSF or the granulocyte monocyte stimulating factors, and lastly macrophage inflammatory factor. Another third situation may also occur where the receptors, they stimulate the mast cell membrane, which releases phospholipase A2 by the breaking down of phospholipids into arachidonic acid with the help of enzymes which are present in this membrane. The phospholipase A2 may undergo through either the lipooxygenase pathway or the cyclooxygenase pathway. In the lipooxygenase pathway, the leukotrienes such as LTB4, C4 and D4 are produced. Always remember that these leukotrienes are very strong vasogenic as well as spasmogenic agents which are 1,000 times more stronger than histamine. And through the cyclooxygenase pathway, prostaglandins are produced such as D2, E2 and F2. D2 is the most abundant prostaglandins produced, which are strong vasodilators, strong glandular stimulators, as well as increases the bronchospasms and mucus secretions. Also remember that these membranes, they produce platelet activating factors which are derived from these membranes, which increases the histamine production, is a very strong vasodilator, very strong spastic agent, chemotactic for inflammatory cells. Let's talk about the third phase or the pathophysiological phase. The second phase leads to the third phase. Let us make it clear that due to the accumulation of the different inflammatory cells such as the eosinophils, lymphocytes, histamine, etc., the third phase takes place, that is the inflammation. Remember that eotaxin is also produced by the epithelial cells, which are an attractant for eosinophils. Let us summarize that eosinophils are attracted with the help of interleukin 3, interleukin 5, T helper 2 cells, cytokines, and eotaxin. We can also divide the type 1 hypersensitivity reaction into the early and the late phases. Early phase is characterized by the vascular and spasmogenic events, and the late phase is characterized by the inflammatory reactions. Hypersensitivity type 1 reactions are divided into three types. First is the localized type, such as allergic conjunctivitis, allergic rhinitis, angioedema, hay fever, allergic asthma, hives or the red patches which is present on the epidermis, and abdominal cramps and diarrhea, which are the allergies against citrus. The second type is the systemic type or the generalized type. Examples are sensitivity to penicillin, anaphylactic shock occurs in this case where the mast cells they degranulate. Injection of peptide hormones also causes such a reaction. The clinical symptoms are hives, erythema, respiratory distress, and cardiac problems. The third type is where both the localized and systemic reactions they take place. Unfortunately, we can treat this type of hypersensitivity reaction by the desensitization therapy, in which a small amount of allergen parenterally is repeatedly exposed to the patient, and the patient starts making IgGs against the allergen, which blocks the allergen towards the IgE. We can diagnose this hypersensitivity reaction by either prick or scratch test and RAST or radioallergoimmunosorbent test. Before we end the discussion, let's talk about atopy. Atopy is a familial tendency to make an increased production of IgE more frequently against common environmental substances to which most population is inert. Remember, it is genetic. Well, that's all for now. Thanks for watching.